Hello, everyone. In the previous lecture, we have studied about transcriptome analysis. And in transcriptome analysis, we studied the best tools to study the transcriptome are microarray and RNA sequencing. In this lecture, we're going to look at the differences between RNA Seq and microarray. So before getting into the differences, we'll have a look at what microarray is. Just remember, for your knowledge, microarray is nothing but a simple slide which contains slots for different samples, and each one of these slots would contain probes onto which you perform the experiment. We'll study about this in detail later on, but first let us have a look at how many different kinds of microarrays are currently there in the market. The first one is the gene expression array. And it's quite evident that these arrays are used to study the differences in gene expression between two conditions. So this studies what? It studies gene expression. The next one, they are genotyping arrays, wherein you study single nucleotide polymorphisms, and this is normally used for variant calling. These two sections we are going to study in detail in our lectures, either this one or the lectures to follow. And the third one is the comparative genomic hybridization arrays. Wherein this is used to study copy number variants. Is the LNC RNA arrays or the miRNA arrays. They can be combined or they can be individually be studied. You have both the options here under the microRNA system, wherein you look at differential expression of microRNAs and differential expression of microRNAs and miRNAs and LNC RNAs. And the last one you have under microarray, they are nothing but methylation arrays, wherein you can study the methylated regions in the genome. And these are often used to study the methylation of the CpG islands. CpG islands. These are the arrays that are currently there in the market and the purposes for which the microarray system is generally used for. We'll now try and understand the principle behind the gene expression arrays. Understanding this will lead to a generalized concept of how the microarray works. Now we'll take control on one side and we'll take treated or infected on the other side. We'll have samples again, like what I have told you. We should ideally have n is equal to 3 was considered earlier, but now n is equal to 6 should be the minimum. That's what I told you. Take 6 samples here, right? Now what you can do is isolate RNA from these samples, from all the samples. And similarly, isolate RNA from all the samples, individual samples. Now what you do is, in the process of conversion of this RNA into cDNA, you label this cDNA with a dye called as, let us say, Psi3. And similarly, Here, in the process of converting this into cDNA, you label this with a dye called as Sci5. Sci5, as everyone knows, is red in color, and this is green in color. Sci3 is green in color. Now what do we do? Now, individual samples. Let us say this is sample number 1 of control, and this is sample number 1 of treated. And now the cDNA that is converted is of sample number 1 here and sample number 1 here, because you have to do it individually. That's what I told you. Then what do? You mix these two samples. You mix these two samples. And this mixture is then put into a slot that is there on your microarray slide. When I say a slide, I've already explained that microarray is nothing but a slide that contains slots for samples. Now ideally you have a slide containing 4 slots for running 4 samples, or 8 slots for running 8 samples. Now look what happens here. Now what do? We'll have to understand what every slot contains. We'll just magnify this, go to a point here, and check that what is exactly there inside each slot. Slot contains spots. When I say a spot contains probes for, let us say, a particular gene. And this is a probe for, let us say, gene A. And this is a probe for, let us say, gene B. And similarly, you have for all the known genes in that particular species of interest, you will have probes distributed across this slot, and they are lying vertically coming out of the slide. Now, when I say a particular spot roughly contains around 10,000 probes for that particular gene. 10,000 probes for that particular gene. Now, this probe is complementary to the sequence of your cDNA that you have already converted in your mix here. Now, what happens? In this RNA, this gene A, for example, let us say this is a gene A, and this is a probe that is there at that particular spot. And if this gene A is expressed in control, and what happens? This cDNA would come and hybridize when your mix that is taken here is actually subjected to denaturation, so that the double-stranded molecules will become single-stranded and allowed to hybridize onto your sample slot. When this hybridizes onto the sample slot, what happens? The one that is coming from the control, let us be more specific, one that is coming from the control, since it is labeled with green, what do you have? This brings in green color attached to it, right? Because there is a Sci3 dye attached to it, that is green color attached to it. Similarly, since the mix already also contains cDNA with Sci5, and there may be some cDNA which may correspond to this particular gene A, this cDNA, among the 10,000 that are present over here, to some of the probes, we'll just draw this here like this, make it a bigger spot, this small one, having 10,000, and let us take one more probe from this. And this probe, let us say now, it binds to this cDNA that is coming from the infected. Let me draw the probe with blue color so that there'll be no confusion. What am I doing? I'm just drawing the probe with blue color here. I'm just taking a bigger spot. Now you have this is a spot that I'm mentioning here, right? And now you have 10,000 probes here. Now, if there is a probe here, that is blue here, that is a probe specific for this gene. Now, what's happening? We take the mix of this, so some of the cDNA of this infected may also come and bind here. So what does this contain? This contains red color, that is nothing but Cy5, and this contains green color, that contains Cy3. Now what happens? Similarly, the same thing happens for all the genes, because this mix may contain all the genes that are expressed in the form of RNA, which are later on converted into cDNA and which are labeled in the form of either Cy3 and Cy5. Cy3 corresponds to control and Cy5 corresponds to infected. Now look what happens. Now let us say, for example, the expression of this particular gene A is higher in control. So what happens? Number of green probes that may bind to the 10,000 probes will be higher. So when this microarray is scanned, and this microarray is scanned for this particular spot that you are looking at for gene A, the color would slightly be towards green. Means this spot after scanning, if you're looking at, will be green in color, slightly towards green in color. What does that indicate? That indicates that the expression of this particular gene is higher in control with respect to that of infected. Once again, that is mainly because you found that there are more cDNA with Cy3 that are going to come and bind to your probes that are already there in number, 10,000 in number. And what will happen? Just because of this binding, the green color will dominate because there is higher expression of this particular gene in control with respect to that of infected. Now we'll have a look at that. Gene B now. Now you have a gene B, and let us say the same thing is happening here. We are taking a spot here. Let us say this is gene B, a spot. This is a spot, right? And out of this spot, what are you doing now? You have probes for gene B. Now this is expressed, let us say I'll draw three probes. We have 10,000 in number. And let us say this expression is higher in infected. Then if it is higher in infected, what happens? It will automatically come and bind here. Right? And it is still expressed in case of control. So you may have some probes binding to the cDNA, which is labeled with Cy3. Now what happens? Just because the expression is higher here, this spot now looks like, if you go to this here, after scanning, it will be slightly towards red. So by looking at the spot, if it is slightly towards green, or depending on the intensity with which it is towards green, or depending on the intensity with which it is towards red, you can differentiate the expression of that particular gene in your control and infected. And you can tell how the expression varies in infected with control, and or similarly the vice versa in control with that of the infected. Now, suppose the expression is similar in both the cases. Now what happens? You have equal number of green binding to the probes as that of equal number of red binding to the probes. Now we have equal number of green and equal number of red bound to the probes. Then the color would be yellow in color. So if you have a yellow color here, just to differentiate this, I'm just highlighting this. You have yellow here, right? This is yellow in color. So by looking at the yellow spot, you can make out that the expression of this particular gene C is equal in both control and infected. Now what happens? The intensity of the color, or the intensity towards green or intensity towards red, will actually indicate the magnitude of expression of that particular gene, one over the other. Means like if it is the intensity towards green is higher, right? It means that it is more expressed in control. And if the intensity is towards red in color, it is more expressed in the case of infected. And depending on the varying intensities, either towards red or green, the intensities are converted into numbers by the software. So here, whatever microarray does is qualitative, means the intensity is caught and then further converted into fold change. What is fold change? Expression of y over x. What is y over here? Is nothing but infected over control. Infected over control. Infected over control. So this intensity, if it is entirely towards green, means the expression will be higher. Slightly towards green means the expression is slightly higher in control in comparison to that of infected. If it's completely towards green means the expression is far higher in control than in the infected. And similarly, the vice versa with the color of red as well. And this is how microarray actually differentiates the expression in the case of infected and control. Summarizing this, we'll go to the next page. This is what I have explained earlier, that normal RNA or the control sample, RT-PCR, green in color, and you have this array here with different spots, right? Let us see one sample. You hybridize, mix, hybridize, wash, and scan. Yellow in color, equal expression of that particular gene. Let us say if it is gene A here, equal expression of this here, because out of the 10,000 probes, equal number of probes would bind to the cDNA that have Psi3, and equal number of probes would bind the cDNA that have Psi5. And therefore, the color would be yellow in color. And if it is slightly towards red, means the expression of that particular gene is higher in this experimental sample. And similarly, if it's towards green, it's higher in the normal. The principle of the microarray remains the same for all the other microarrays that we have discussed earlier. The take-home message here is that the qualitative signal that you have taken here is basically converted into fold change, FC, fold change. And what did I explain? Fold change is nothing but y by x, where y is the expression in experimental that is towards red in color. Let's put it this way, towards red in color. And similarly, where x is nothing but the expression in control, which is green in color. Is green in color. So if the spot is green, naturally the value of x would be higher than the value of y. So this value would be less than 1. And similarly, if the spot is red in color or towards red, the value of y will be higher than in x. And therefore, the expression would be greater than 1. Now, we'll try and understand how microarray differs from that of RNA Seq. We'll try and understand how microarray differs from RNA Seq. In the RNA Seq experiment, we generate data depending on the type of experiment we perform. We have already discussed under RNA Seq the whole transcriptome, wherein we can find out the expression of mRNA, LNC RNA, and circular RNA. We can also, under RNA Seq, simply do mRNA Seq, wherein the RNA specifically is used along with the oligo-DT probes to isolate mRNA, and this mRNA is used for sequencing. And this data can only be used for differential expression of genes. Call them as DEGs. Then we can also do RNA Seq. Today, for our ease of understanding, we will take mRNA Seq as an example and move forward. We'll again have two groups here, like we have done in the previous lectures, control and infected. Under each of these groups, we'll have 6 biological replicates. For each of the replicate under mRNA Seq, we had already seen that we need to generate 3 GB of data, which corresponds to 40 million reads of 2 into 75 base pair. That is what we had looked at. And this data basically has this format, which is called as FASTQ. And each read runs into 4 lines, and the second line is nothing but the actual read. When you're looking at the read, there is no information to which gene that particular read corresponds to, or which part of the genome that particular read corresponds to. So now what we do is, we take the reference. When we call it as a reference, it is a FASTA file which runs entirely across the entire genome and is completely annotated. When I mean annotated, each and every part of the genome is clearly known or defined. This to this is gene A, and this to this is gene B. This to this is gene C, and this is the intergenic region that is here, and this to this is gene D, let us say. Now, for this sample, you have a FASTQ file that you have generated, and which has reads in this. These reads are now mapped onto the reference. The word is actually called as mapped onto the reference. When they are mapped onto the reference, some of the reads would go and map here. Some of the reads would go and map here to this. Some of the reads would go and map here. And some of the reads would go and map here. Now what happens? We count the number of reads that are mapped. For example, the number of reads that are mapped are 3 here. And the number of reads that are mapped are 5 here. And the number of reads that are mapped are 4 here. And again, the number of reads that are mapped are 3 here. Now, the same thing is again done with respect to that of your infected. We take the reference, and then again, the same genome, which is completely annotated, is again used for mapping the reads of your infected for each sample separately. Take this sample again. This is the FASTQ file, and the reads now are mapped here. And let us say here, the number of reads that are mapped are now 6 in number, and the number of reads that are mapped here are 3 in number, and the number of reads that are mapped here are just, let us say, 2 in number, and the number of reads that are mapped here are again 6 in number. So let us write the numbers here. We have 3 here, 2 here, and 6 here. Now, initially looking at the counts of these reads, we call them as raw counts. I'll tell you why we call them as raw counts. Initial look of it tells us that for gene A, the number of reads are 3 here, and the number of reads are 6 here. And if we look at it, it means that for this particular gene A, the expression is higher in the infected. It means the gene has got upregulated in the infected. And if you look at gene B, it is 5 here and it is 3 here, and the expression has come down. It means gene B has come down, and therefore it is down-regulated. So the first look of it, by looking at the raw counts, it tells us that the counts in the infected, if they are higher than the counts in the control, it means that particular gene is upregulated, and the counts in the infected are lower than the counts in the control for a particular gene, then it means it is downregulated. However, we will have to normalize are raw counts, which we shall discuss in detail later on. The point that I want everyone to understand here is, in RNA Seq, we will be getting the counts directly, and therefore it is quantitative in nature. Quantitative in nature. And if you want to check the fold change for a particular gene, let us say for gene D, and if you call this as Y and call this as X, and if this is Y by X, we will be having what? We'll be having 6 by 3, and that is nothing but 2. Means there's a twofold increase in the infected with respect to that of your control. And that's how you estimate the fold change for a particular gene, that is fold change for gene B. However, a word of caution, we do not use raw counts directly for our calculation for fold change. We have to normalize our reads for gene length, library size, and composition of RNA. Composition of RNA. These are the 3 things that we need to normalize for, and we'll discuss this in detail when we discuss RNA sequencing in the next lecture. Thank you.